The overall goal of this procedure is to create polyacrylamide gels that can be used in both Invadopodia and traction force assays to study the invasive and contractile properties of cancer cells. This is accomplished by first activating the 14mm cover slips in the wells of glass bottom dishes to ensure that the polyacrylamide gels are bound to the glass. Next, polyacrylamide solutions for both assays are prepared to yield gels with different rigidities or mechanical properties. Then, the Invadopodia assays are prepared with a thin layer of fluorescently labelled extracellular matrix to detect degradation. Finally, the assays are seeded with cancer cells for overnight incubation, followed by fixing and staining of the Invadopodia plates and live cell imaging of the traction force plates. Ultimately, fluorescence microscopy is used to image Invadopodia and extracellular matrix degradation, while fluorescence and wide-field microscopy are used to image microspheres and cells respectively for traction force calculations.
Demonstrating the procedure will be Rachel Jerrell, a research assistant from my laboratory.
To prepare glass cover slips for polyacrylamide gels, begin by using low lint wipes to clean 12mm cover slips. Use tweezers to hold 12mm cover slips and 35mm glass bottom dishes containing 14mm cover slips and pass them through a Bunsen burner flame. Treat the microwells with 200 microliters of 0.1N sodium hydroxide for 5 minutes at room temperature. Then aspirate and air dry the microwells for 30 minutes. Next, under a fume hood, use a glass pipette to apply